So this is the second video of the intro to innate immunity. So one thing we want to talk about for this section is how do those macrophages, dendritic cells, neutrophils of the innate immune system, how do they recognize pathogens and notice that these are bad guys that we need to take care of. So the way that they do that is through several different receptor types. Most famous among these is probably the toll-like receptors. The toll-like receptors are um, the toll-like receptors are so named because they have similar motifs to the toll receptors in flies. But there are other receptors too, immunoglobulin-like receptors and some G protein coupled receptors like the mannose receptor and the glucan receptor, as well as the scavenger receptor, which is associated with uh, complement. So we've got lots of receptors on this macrophage, and when it senses something that is common in one of these pathogens, it would react to it by uh, a couple of processes. One is phagocytosis, and the other is releasing cytokines to recruit other innate immune cells to the area, as well as start the inflammatory process. So, um, these sensory cells, they could be macrophages, neutrophils, dendritic cells. They could also these receptors can be on the endothelial lining of a blood vessel. They could be on in any of the cells of the tissues, and and they would be recognizing these common motifs like single strand RNA, for instance, or lipopolysaccharide. So so once the macrophage recognizes that we've got a pathogen, a bad guy, the first reaction of the macrophage would be to trigger endocytosis. So it's going to gobble up these bacteria. Hopefully, it's going to be able to gobble them up. Um, if not, then it needs to recruit some help. So it's going to release some cytokines and chemokines. So before we had several different receptors, but downstream of all of them is going to be uh, cell signaling pathways. If you haven't had cell biology yet, um, let me know. I've got quite a bit on cell signaling in my cell biology notes, and I can get you caught up pretty quickly. Um, but um, but there's going to be some cell signaling downstream of these receptors and the release of cytokines and chemokines. So when the endothelial cells sense these cytokines and chemokines, they're going to have uh, a cell signaling of their own, which is going to cause them to change their morphology. So if it's the endothelial lining of a blood vessel, then one of the downstream results to these cytokines is to have some fenestration, some becoming more permeable, more leaky, and of course the pressure inside of the blood vessel is greater than that in the tissue. The contents of the blood vessel will leak out. So this is going to explain three of the four hallmarks of inflammation: the rubor, calor, and dolor. I'm sorry, torpor. Rubor, calor, and torpor, or redness, heat, and swelling. Because if you've got uh, the contents of a blood vessel leaking out into the tissue, it's going to cause it to become swollen. The blood is warm. That we are endotherms, and so it's going to cause heat and redness. And so the redness, the heat, and the swelling of inflammation are all caused by this increased permeability of the endothelial lining of the blood vessel. Some of the cells that are going to extravasate and move into the tissues, such as the neutrophils and the monocytes, they are going to release other inflammatory mediators, such as substance P, and that is where we're going to start getting the pain or the dolor. So rubercalor and torpor are all due to just the becoming more permeable of the blood vessels, and then the dolor. Did I say? Oh, okay. Rubercalor and torpor are due to increased blood in the tissue, and torpor is due to some of the things that the granulocytes release cause pain. And this is all going to help with dealing with these pathogens, because if you've got an infection in the bottom of your foot, you're going to want to stay off of it, and so the increased pain is going to keep you from walking on that infection. Meanwhile, the swelling is going to prevent the bacteria from being able to get into the blood vessels, and the heat may be enough to kill the bacteria. Increased temperature will kill bacteria. So all of these inflammatory mediators are all going to come from that original response to these common motifs of bacteria sensed by those sensory cells like macrophages. So one of the cells that may come to help would be a natural killer cell. Natural killer cells are myeloid lineage, but I'm sorry, they are lymphoid lineage, but they are part of the innate immune system. They're lymphoid lineage, same as the B cells and T cells, but they are part of the innate immune system in that they have granulocytes that they will release. So their mode of action is very similar to that of T cells. They're going to release chemicals that will disrupt the cell membrane of, say, like bacteria or a virally infected cell of your tissue, and those chemicals will disrupt the cell membrane and cause lysis of the cell. So one of the things that we want to talk about this semester is the interaction between the B cells and T cells with the innate immune system. So how are antigens and antigen receptors produced? We'll talk about that in chapter 4. And what are the downstream signaling processes? So chapters 5 and 6 for B cells, and 7 and 8 for T cells. And we'll talk about how lymphoid cells ultimately lead to clearance of the pathogen, as well as memory for the next time that you're infected. So along that line, the B cell receptor is 4 proteins, 2 different gene products, each of 2 different gene products to create a quaternary structure of 4 different proteins to create a globular protein that has a very specific antigen recognition and binding site. T cell is a little bit different. One is that it's only two proteins, so it is 2 gene products, just like with the B cell receptor, but but there's only going to be one each of those. And again, a variable region or antigen binding site that is unique. And we will talk about how that is produced in chapter 4. And then how they are going to do their mode of action, we're going to subsequently cover, like what is the cell signaling. The response to an antibody or a T cell receptor are different. They have unique modes of action. And that's because with an antibody, you're looking for a native protein. And so the antigen may be globular, and the actual epitope recognized by the antibody, it doesn't have to be sequential amino acids. They could be amino acids that are very far apart from each other in the primary structure of the antigen, while being right next to each other in the tertiary structure of the antigen. And this is very different from the T cell structure or T cell recognition spot, because the mode of action of T cells is different than antibodies. Antibodies are released and then are going to bind to proteins maybe in the milieu of the blood or on the surface of a protein. And so they need to be able to spot that epitope in its native form. On the other hand, with T cells, after that bacteria gets engulfed by that macrophage, it's going to chop up that bacteria, and it's going to chop up all the proteins of that bacteria. And so the portion of the protein that is actually presented to the T cell receptor is actually primary structure, not tertiary structure. So it could be something that is only seen deep in the middle of the protein, and wouldn't actually be on the surface for an antibody to see. So after it chops it up, it's going to present it on a MHC, or major histocompatibility molecule. And it depends on whether we're looking for attacking virally infected cells, or in the case of a macrophage gobbling up a bacteria, some exogenous protein, which MHC it's going to be presented on. So if it's a virally infected cell that is presenting this protein, it's going to present it on MHC class 1, and that's going to activate CD8 T cells. Whereas if it's something that was engulfed and chewed up, it's going to be presented on MHC class 2 and activate CD4 T cells. And we're going to have a chapter on each of those two scenarios this semester. So when a B cell or a T cell creates its receptor, the B cell receptor or the T cell receptor, it does it 100% randomly. So it's just rearranging DNA until it gets a B cell receptor or a T cell receptor. So we're going to need some sort of tolerance mechanism, because we don't want our B cells and T cells attacking our own proteins. And so that's going to happen in the case of B cells in the bone marrow, in the case of T cells in the thymus. While these things are developing, we are going to have tolerance or negative selection that gets rid of anything that is autoreactive, hopefully.